Continue with the previous video, that is Selaginella morphology and the sporophyll structure and classification. The continue with that, we'll talk about the anatomy of the root, stem, and the rhizome regions. So usually coming to the root TS of root have the three distinct region, that's the cortex and the stellar region. The first thing is the figure is showing the outermost region that consists of the epiblema region. The epiblema region having the root hairs. Below that you will find the larger that's hypodermis region, and center cortex will be there. Usually they consist of the metaxylem and the protoxylem region, endodermis region, and the phloem region. So usually epiblema is a single layered made up of tangentially elongated cells, and the cortex is usually homogeneous and consists of many layers of the parenchymatous cells. Even sometimes they may be a thick, or it may be sometimes varies and have sometimes they may be a mycorrhizal association. And below that the cortex consists of the endodermis region. The center, the stellar region will be there, and usually the center part that consists of the protostele, that consists of two to three layers of the parenchymatous pericycle, and xylem will be there. Coming to the anatomy of the rhizospore, this is a special characteristic of the Selaginella. So usually almost resembles the characteristic of the root, that consists of the epiblema, cortex, and the stellar region. This is the figure is showing the structure of the rhizospore, anatomy of the rhizospore region. That's the epiblema is the outermost layer. Below that you will consist of the thick layer of cortex, that's the sclerenchymatous cortex there, and also consist of the parenchymatous layer will be also there. And in the center you will find the endodermis, with that the stele, that's a protostele will be there. And usually the protostele that consists of the metaxylem and the protoxylem in association with the phloem. The coming to the anatomy of the stem, the internally the stem is more complex than the root. The anatomy of the stem shows the various regions or the various structure. So the usually just like the root, they have also the three regions, that's the epidermis, cortex, and the stellar region. The figure is showing the outermost layer having the cuticle. Stem have the cuticular region. Below that they consist of epidermis, and also the epidermis is the outermost layer, that's the parenchymatous, and usually they are interpreted with the stomata and also the hair. Or cortex usually the composite of the parenchymatous cells without the intercellular spaces, and usually partially the sclerenchymatous layer will be there. The xerophytic species cortex comes from thick walled cells, and innermost layer of the cortex forms the endodermis. Endodermis cells are elongated due to the growth of cortical cells, and usually during their development radial elongated there, and also they became the more cells of filament. And the important region, that's the endodermis cells, have the specialized structure called as a tuberculae. These tuberculae are the distinct features of the Selaginella stem. Sometime maybe in examination question will be raised. So where do you find tuberculae? So usually tuberculae is present in the stem or the endodermal region of the Selaginella. And sometime in xerophytic species this tuberculi may be absent, and the center portion consists of the stele. Stele is a distinct region, that's a monostelic and distelic and the polystelic region will be there. And sometime maybe it varies how actually the figure is showing the regions. Sometime in case of the different species, different types of stele will be there. There are circular ribbon like structures will be there in a stellar region. The central portion is generally referred as a stellar region, and pericycle will be there. And xylem is usually monarch or diarch in nature. This is the electron microscope shows the region of the. So this is the electron microscope region of the center portion of the Selaginella stem. So that continue with the chapter, that is the reproduction. So usually the sporophyte of Selaginella produce vegetatively as well as asexually, the vegetatively by the fragmentation and by the root tubers and also by the resting buds. The fragmentation is simple method of vegetative reproduction. Just cut down the main branches and they will develop into new one. And the root tubers, these are the special underground root tubers will be developed near the rudimentary scales and they will develop into new ones. During the unfavorable condition, this type of vegetative reproduction will be there, and usually and sometimes they may produce the dormant or the resting buds will be produced. They will cut off and to produce the new ones, or even sometimes maybe a rhizospore through that also it may be. And yes, actually, the yes actually by means of spores, and there they produce two types of spores. That's the megaspore and the microspores. So megaspore is produced from the female gametophyte, and microspores produced from the male gametophyte. The sporangia is distinct. That is a dimorphic. That why? Because dimorphic, because they produce the micro and the megaspores separately. So that's why megasporangia and the microsporangia will be called. And usually microspores produced from the microsporangium, and usually 1 to 4 megaspores will be producing. So megas means larger sized and micro means small sized one. So this is how the structure of the sporophyll. They consist of the strobilus, and the single is called strobili, and the varies of this strobili is varies 5 to 7 centimeter in nature. And usually this is the photograph is showing the strobili structure. So strobiles in some of the species Selaginella, both micro and megasporophyll will be formed in the same strobili, but in some species it may be varies. So in case of Selaginella Christiana, there is only single megasporangium will be there. In case of the Selaginella equenta, one side of the strobili bears the only megaspore, like that the structure will be varies. In some time they may be in monosporangiate, the both megasporangia are born in a separate strobili. So these are the some pictorial representation of the sporophyll or the strobili. The base region that consists of legules and central axis will be common. The tip region consists of that the microspores and microsporangium will be there, and the other where actually microsporangium is protected the leaves, that's called microsporophyll, and below that we will find the megasporophyll that consists of the megasporangium. Coming to the structure of the mature sporangium, so usually they are stalk structure and two layers sporangial jacket will be there. Cells of the outer jacket layers are elongated, contain the chloroplast, and micro and megasporangium different shapes and the size structure. So coming to the microsporangium, the pale yellow in structure and usually oval or spherical shaped in structure, and usually they bears on the microsporophylls. Usually they are surrounded 2 to 3 layers of the jacket and one layer of the tapetum enclosing of the microspore mother cells, and usually they produce to 400 to 2000 of the microspores produced in a microsporangium. And the megasporangia, they are pale greenish yellow in color, and usually sometime they may be yellow or orange in color, and usually they only produce 2 to 3 layers of jacket layer will be there, and only 4 megaspores will be produced in a megasporangium. And when the produce microspores and megasporophyll, and going to the reproduction, zygote will be formed. The gametophytic structure start with the formation of the zygote. So usually spores mother cells gametophytic generation, as mentioned earlier, the Selaginella is a heterosporous. That means they produce the two types of spores, that's one smaller and the larger size spores. The smaller size are called microspores and larger size one called megaspores, and usually these are developed from the microsporophyll and the megasporophyll, that's the male and the female gametophyte. And when they get germinate, so usually first microsporophyll undergo to 13 cell stages, and later it will be develop, start the follow the different stages of the development. And this is the actually how the pre megasporangium is the formed in the sporogenous cavity. Coming to the structure of the mature antheridium, so this is the structure of the antheridium. They consist of usually single jacket layer and goes with the centrally androgonial cells, and usually they produce the more than 100 to 250 antherozoids, and they undergo to metamorphosis, and usually these antherozoids are the biflagellated one. And coming to the archegonia, they consist of the neck canal cells, and usually two neck canal cells, and the eight cells in the two rows will be there, and four terminal cells neck projection the surface will be there. Fertilization usually takes place after the megasporium has fallen into the soil, and usually just before the fertilization, neck of the archegonia substrate into the form a passage for the entry of the antherozoids. When coming in contact with the antherozoids and the egg cells, the fertilization will be takes place and the zygote will be formed. This is how actually the complete structure of the life cycle of the Selaginella. The first stage that represent the sporophyll of the plant, second one represent the strobilus, and the third one is represent the sporophyll, fourth one represent the sporangium, and fifth one stage is represent the microspores, and sixth one is a male gametophyte, and seventh one is the spermatozoites or the sperm, and eighth is also represent the sporophyll, and ninth one is the sporangium in nature, and tenth one is a megaspore, and 11th one will be female gametophyte, 12th one that is the archegonial in shells, and 13th is the egg cells, and 14th will be zygote will be formed, and 15th will be young sporophyte. The 1 to 15 different level of the stages is there. So usually through that the microspore and the megaspore undergo to the germinate to form the structure. So this is the structure of female gametophyte, and this is the how actually microspores will be developed inside the cavity. So these are the microspores, that's they produce the antherozoids, and this is the complete structure, and this is the megasporophyll how actually the develop in case of the Selaginella. So this is the usually where actually the dark blue indicate the haploid stages and light blue indicate the diploid stages. The usually the sporophyte or sporophyte region is the diploid, and the gametophyte is haploid in nature. So this is the alternation of generation or the life cycle of the Selaginella. So this is the and complete account of the Selaginella. So in the previous video we are discussed the morphology, habitat, and structure of the Selaginella. In this video we are have talked about the Selaginella anatomy of the stem, root, and the rhizospore that continue with the reproduction in case of the Selaginella, and the structure of the microsporophyll and the megasporophyll and life cycle of the Selaginella. So I hope you have understand some concept of Selaginella. So in the examination you have a question on the generally especially the structure of the microsporophyll or megasporophyll, or explain the structure of antheridia, archegonia will be asking, and especially in the rhizospore region, the anatomy of the rhizospore is a special characteristic, and also the stem anatomy. So you have a chance to explain for the 5 to 6 marks. You have to write the neat label diagram and explain the structure, and maybe sometime maybe ask for 1 marks what is a ligulate, where do you find the some structure in case of the Selaginella, what is a strobili. So like that there will be a questions will be there in the examination. I hope you will get the point, and later we will discuss the other topics.